Okay, testing my mic. Testing. Okay, so my mic has sounds. Okay, so already 8 o'clock. So now I will continue the class. So today class is semester 3, biology about chapter 17. So today's topic will talk about gene expression. Okay, gene expression. So this topic also maybe is totally new to you. You never learned before in your level. So gene expression actually in another name we can also call it essential dogma. So what is it? Later we know that. Okay, so for today we will cover this all lah. These all topics. So before we go to the transcription and translation, we need to know the breeder and tenter experiment and genetic code. Okay, so actually what is this is the last experiment lah. The experiment number 5, kelima. Experiment number 5 that you learn in this chapter. So this chapter altogether contain 5 experiments you need to learn. So 3 experiments are included in DNA replication, right? To to know that DNA carry genetic material, and another experiment is to determine the model of DNA replication. And then this experiment is to determine the expression experiment. It need to just want to confirm that our gene can express the protein. How our gene can express our protein. So this experiment are conducted by these two scientists, Bieder and Tatum. So this experiment I can say that is also important for the scientific field because this experiment gives the conclusion about this. So it gives conclusion about this one gene, one polypeptide hypothesis. Meaning that one gene will produce one polypeptide. So this experiment is to prove the gene expression. So actually for the scientists at the beginning, the scientists actually don't know. Mereka pun tak tahu apa itu gene expression. Mereka pun tak tahu sama ada itu our gene only carry genetic material, but they don't know is it betul tak gene boleh express to produce protein. Betul tak? Mereka tak tahu. Semua saintis pun tak tahu. Tak ada scientific evidence yang kukuh. So, yang ni, eksperimen ni, dia buat a proof lah. Dia proof this experiment to prove that our gene material can be expressed to form the amino acid. So, how it form? It gives the one of the fungi which we call as Neurospora crassa. So this fungi actually is the fungi when your bread is rosak. Kalau roti awal rosak kan dia ada hitam-hitam semua ni kan. So yang ni hitam-hitam ni adalah fungi lah. One of the fungi species that is we call as Neurospora crassa. So this is the main species to determine the DNA mutation. So why that two scientists choose these fungi rather than choose bacteria or choose other type of cells? So why it choose this? So normally there are 6 reasons lah. So this first reason is to easily to culture in minimum medium, meaning that the culture of this fungi does not require many complex nutrient. Only just minimal nutrient, meaning that just only certain basic nutrient, then it already can survive. And then it is very small and breed in large number in small space. So you see this breed, so you can see that there are the population of this species Neurospora crassa is very high lah. Although you see that breed is limited space, but it can produce in very large number. And then it has the short life cycle. The life cycle is only 10 days. So you can get the results very quickly lah compared to other types of cells. And another one is it can be exhibit diploid and haploid stage in the life cycle. So this one actually you will learn about the fungi life cycle. You will interesting. You can refer to the Campbell book one. Campbell got explain about the life cycle of this fungi. So you will not you don't interesting. Never mind. You don't need to explore it. You just want to know that this stage because occurrence of this stage so that the scientists can be easy lah can make a comparison between these two stages. And then few chromosome gene local are easily to determine. So one of the important is the chromosome of this fungi is very little. So that the scientists can easily to study the gene local of the chromosome. So compared to human contain 23 genes, right? Not 23 genes, 23 human got 46 chromosomes. So you use human normally take times, right? And normally you want to study the chromosome already take times already. Kalau awak tak buat eksperimen, just study the chromosome pun dah banyak masalah. So awak guna ni, so we can easily to determine the gene sequence of this species. And then it can produce a spore asexually. So it is asexual organism. So it can rapidly to produce more and more daughter cells lah. So this is the intro why we choose this fungi. Okay, now we go to the procedure. Okay, so how this experiment conducted? Okay, so firstly we need to culture the neurospora cells. We need to culture it. We need to grow it. Kami nak banyak neurospora cell. We just culture it in the complete medium or defined medium lah. Okay, after we culture the neurospora cell, dia ada banyak number. Okay, then we export these cells in X-ray. Kami buat X-ray dekat sel ni. Why? Because we need to induce mutation. We need to break down. We need to not destroy lah. We need to mutate the gene sequence of these cells. Kami tak mahu normal punya. Kami nak yang mutant punya, yang tak normal punya. So why it need to reduce the induced mutant? If we use the normal one, actually we cannot make a comparison. If we use normal one, mean that this fungi can be express the normal type of polypeptide change, right? For normal type of protein. So we cannot make a comparison. So if we induce mutation, meaning that different type of the cells will have the different type of mutation lah. Normally. So if we induce mutation, so normally most of the cell will die. Only the some of the cell can survive only. So want to see lah what kind of mutation occur, so that kind you don't need to know. You just want to know that only the surviving cell after you do a mutation, you will found that only some cells can be survived. Yeah, others will be dead. And so the surviving cell form a colony of genetically identical cells. And then after that, we will culture again. We use these cells, these already mutated cells. We culture again in the minimal medium. We culture again in the minimal medium, same as this one, as the first step. In the first step, we culture in the minimal medium, complete medium lah. Complete medium means that there are extra nutrients. But for now, we culture in the minimal medium, meaning that the nutrient content is very minimum. Sangat cukup je, ngam je, cukup je. Kami tak bagi extra punya. Macam awak makan makanan lah. Saya hanya bagi awak biskut je. Awak sangat lapar, tapi saya hanya bagi biskut awak. Saya tak bagi nasi. Saya tak bagi ayam, daging, semua tak bagi. Saya hanya bagi biskut je. So, to see that whether the cells can be survived or not. So, if you take the mutated cell, go in the culture in this minimal medium, they will found that the cells cannot grow. Cells that did not grow were identified as nutritional mutant. So only the cells that cannot go, we can identify as the nutritional mutant. Meaning that we want that cells. Kami nak cells tu. Why? Because it acts as nutritional mutant. Why? What is mean by nutritional mutant? Because as I mentioned before, if the normal Neospora crassa fungi, it can easily go in minimal medium punya. Sangat senang je. Dia akan go punya. Memang dia akan go punya. Walaupun dia sangat lapar, tapi awak bagi biskut je, saya bagi biskut je, awak pun boleh survive lagi. But when they put the mutated cells into the minimal medium, they found that the cells cannot go already. So meaning that these cells is already have the nutritional mutant, meaning it require the specific nutrient to grow. Dia kena ada satu specific nutrient baru boleh tumbuh. Kalau tak ada, dia memang tak boleh tumbuh. But what are that nutrient? They don't know. So that is why they need to do an experiment, further procedure. So only the cells that cannot form colony, they will choose. Kalau yang tak ada koloni dekat minimum medium, meaning that although it is undergo mutation, but it is not the nutritional mutant. So the mutation maybe is maybe take part at anywhere lah, tak tahu mana lah, tapi yang kami tak nak lah. Kami tak nak punya. Kami hanya nak yang tak boleh go in the minimum medium. So if you know that which colony cannot go in the minimum medium, we use that same colony again, go into the different amino acid culture. So meaning that cells from one nutritional mutant colony were placed in a series of vials, each containing minimum medium plus one additional nutrient. So for this experiment, they use amino acid. So meaning that they will go to the the same colony, they will go onto the different 20 tips. Maksudnya dia akan satu colony ni, dia akan bahagi kepada 20 test tube. Dan ni adalah 20 test tube. Why 20 test tube? Because we have 20 amino acid. We have 20 amino acid, right? We have 20 types of amino acid. Maybe I use a different color lah. 20 types of amino acid, right? So when you use this 20 types of amino acid, meaning that each tube, all these tube, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it will be separated into these 20 types of amino acid test tube, and then each test tube only contain minimum nutrient and one type of amino acid plus one type of amino acid. Only one type. Maksudnya kalau ini adalah lysine, yang lain tak ada. Ini hanya lysine, ini hanya valine, ini hanya arginine, ini hanya aspartic acid, ini hanya methionine, ini hanya dia lagi lah 20 jenis lah. All of this. And then they need to find lah which type of amino acid can make the cells growth. Finally, they found that only the medium with the arginine acid, amino acid can grow. Mereka buat semua tak boleh go. But only the arginine, with the presence of arginine amino acid, then it will go. So why? Kenapa? Why? So they need to know why. So later they will do this as explanation. You can pick yourself. Okay, so now they will go to further again. They will find that only arginine. Why arginine boleh survive? Kenapa arginine? Scientist pun nak tahu kenapa arginine. Why cannot methionine? Why lysine tak boleh? Why methionine tak boleh? Why aspartic acid tak boleh? Hanya arginine je. So this they will choose the they will choose based on the mutant, meaning that from now they were to study the mutant class that will or maybe occur in that gene. Just now we don't care about the mutation. What type of mutation they don't care. Other mutation okay je. But after that they found that arginine is the best only the amino acid that can go. So they to found the type of the mutant. What are the mutation occur in that gene? What are the mutation occur in that gene? And then they finally conclude that there were only this gene, the gene only have 3 type of mutation only. Class 1, class 2, and class 3. So which type of mutation you don't need to know, you don't need to know it. You just want to know that they conclude that got 3 type or 3 classes of mutation will be occur at there. Okay, so meaning that, and then they further study again. So based on this mutation, when this is the wild type is the normal one. If normal one, normally you go in the any medium, they will go one. Normal type. And then this one is class 1 mutant. So class 1 mutation, all of this mutation, when we go in the minimum control, memang dia tak ada koloni punya. Tak ada koloni punya. Same as this result. Kami nak macam ni punya. But however, when they are medium, when they add with ornithine, kalau tambah dengan ornithine amino acid, they found that class 1 mutation dia ada koloni. Dia ada koloni. Kalau add amino acid, dia ada koloni. Tapi yang ni dua tak ada. Ini dua tak ada. And then when you add citrulline amino acid, they found that class 1 and class 2 ada koloni. Class 3 tak ada. And then when they add arginine amino acid, which we control, why we add a control? Because we already know lah. We already know that all of the mutation, they will go when the arginine amino acid is present. So confirm or both class 1, class 2, class 3 mutant can go. So scientists want to know why class 1 and class 2, class 1 and class 2 boleh go kalau ada ornithine acid and citrullic acid. But for class 1, class 1 only ornithine acid can go, class 2, class 3 tak boleh. And then citrulline, class 1, class 2 boleh go, tapi class 3 tak boleh. Kenapa? Scientist pun ada question mark dekat sini. So dia pun tak tahu. So they further study again. So they further study what? They further study the gene sequence of this species of this Neurospora crassa. So they study the gene sequence of the Neurospora crassa. So they further study the gene sequence about this. So they found that there are 3 main genes control it. So what does this mean? So they found that there are 3 genes we naming as gene A, B, C, gene A, B, and C. So these 3 genes, will be expressed to form enzyme A, B, and C. And then they found that arginine is the main amino acid that contribute the growth of the Neurospora crassa. Kalau ada arginine, dia baru boleh go. Kalau tak ada arginine, memang fungi ni dia tak boleh go langsung. So this is the key growth factor of this fungi. So that is why that when we add arginine actually does not affected by this origin. So because arginine is the final key growth factor, dia adalah final key growth factor that can make the this Neurospora crassa go. You can imagine say that it like glucose molecule lah for us. Kami memang nak makan dia. So makanan dia adalah arginine acid. Memang arginine acid is a key control factor. Ada dia je boleh go. Kalau tak ada, memang dia tak boleh. But this is of mutant type lah. Because it already undergo mutation. Karena dia sudah undergo mutation. But normally gene mutant type. So actually in normal one, all type of amino acids still can survive. But this one is already mutant. Mutant meaning that our gene sequence is already be affected. Be affected already. So what does this gene can be control the growth of this fungi? So for wild type, when the gene is all are the normal, when the genes all are normal. So if you see the wild type, wild type one, when the genes all are the normal, so gene A can express enzyme A, B, and C. So gene A is it will produce enzyme A. Enzyme A is the responsibility to convert precursor. Precursor meaning is other types of component lah. Because we don't need to learn very detailed about the experiment. So if you want to learn detail, actually it's very detailed lah. But for in Campbell, dia tak bagi detail lah. So precursor is other type of organic molecule, any type of organic molecule or any type of amino acid. So when got any type of amino acid, when got the presence of enzyme A, it can convert or take the reaction to the precursor to change it to ornithine. Dia boleh tukar kepada ornithine. So meaning that gene A, not gene A lah. Enzyme A is produced from the gene A, right? Gene A is produced from enzyme A is produced from gene expressed by gene A, right? Okay, so gene A, kalau ada gene A, dia akan precursor akan tukar kepada ornithine. Presence of enzyme A will change the precursor to become ornithine. So what kind of reaction you don't need to know. Apa reaction tu you don't need to know. And then ornithine kami ada another gene, another enzyme is enzyme B. Enzyme B. Okay, this enzyme is responsible to change the ornithine become citrulline. Dia boleh tukar kepada citrulline. Okay, and then this enzyme C expressed from the gene C, it is responsibility to change citrulline to arginine. So arginine is a key factor. I already explained it. So this is when in normal condition. So if when the enzyme the gene is mutated, meaning that it cannot produce the enzyme already. It cannot produce the normal function enzyme already. So in class 1 mutation, meaning that our enzyme A is already mutated. Enzyme A kami dah mutate. Maksudnya our precursor molecule cannot precursor other types, any types of amino acid yang selain daripada tiga ni, yang lain like methionine, lysine, glycine, aspartic acid, all of these, they cannot change to ornithine already. So that is why that when you do the experiment, you will found that only arginine boleh grow only. Yang lain semua tak boleh grow. Kenapa? Because of this. Because our gene A, enzyme A is already degraded. Kalau dia dah degraded, memanglah already dia tak boleh function. So the precursor already cannot produce ornithine. But why so that is why that when for class 1 mutant, when we plus ornithine plus citrulline, it also can go. Because enzyme B and enzyme C still function. Enzyme B still can change the ornithine to become citrulline. So when we add ornithine, memang dia boleh tukar kepada citrulline, and then citrulline boleh tukar kepada arginine. So that is why that when we add ornithine and citrulline, it still can go. The fungi still can go. Okay, so this is for class 1 mutant. And then for class 2 mutant, okay, class 2 mutant is a mutation occur in B. Okay, in gene B. So maksudnya gene enzyme A and enzyme C is function normally, but enzyme B is mutated. What does mean? Meaning enzyme B tak boleh function lah. It already can tak boleh function because our gene B is already mutated. So our enzyme B is cannot function already, meaning that we cannot this microfungi cannot change ornithine to become citrulline already. Dia tak boleh tukar ornithine kepada citrulline lah. So kalau tak boleh tukar, maksudnya when you add citrulline, kalau kamu tambah citrulline, so tambah citrulline pun, dia pun tak boleh go already. Because our enzyme B that very responsible for ornithine to citrulline is already blocked. Dia already blocked already. It already blocked. So, kalau awak tambah banyak-banyak pun ornithine, dia pun tak boleh tukar kepada citrulline lah. Kerana dia sudah block lah. Dia macam block lah. Sudah halang dia. So that is why when you add ornithine, dia tak boleh go lah. But you add citrulline, yeah, it still can go because enzyme C still function normally. It can change the citrulline to arginine. Okay, this is about class 2 mutant. Now we proceed to class 3 mutant. So this one is about the enzyme C is enzyme C is cannot function already. Gene C is mutated. Our gene C is mutated now. So mutated meaning enzyme C is cannot function lah. Dia tak boleh function lah. So dia tak boleh function meaning that this one is blocked lah. Our citrulline cannot change to arginine already. So that is why that you add ornithine, you add citrulline, or you add other type of amino acid, still all cannot go. Why? Because the enzyme C is already cannot function. It already blocked all the way already. So that is why that you add the arginine amino acid, dia boleh go. Yang ni semua tak boleh. So that is why that when you add arginine, dia boleh go. So this is about the class 1, 2, 3 mutant. So this is the result it proves. So from here, what do you see? From here, what do you see? So from here, you will see that one gene will produce one enzyme, right? Satu gene dia akan produce satu enzyme. We can found that enzyme is a key role lah for this reaction, and then we found that one gene can produce one enzyme. So actually, there in beginning of the hypothesis, before we go to this conclusion again, actually the conclusion made by the leader and dictum is one gene, one enzyme hypothesis. One gene, one enzyme hypothesis. Before it changed to one gene, one polypeptide. So it later they eliminate this hypothesis lah. So why they eliminate? Because not all protein is enzyme. Not all protein is enzyme lah. Not all of the protein is enzyme. Maybe some protein is act as the antibody, act as the transport protein, all this lah. So we cannot say that all of the gene can only express enzyme only. Bukan. Actually, gene is express the protein molecule. Protein. Bukan enzyme. Bukan enzyme. Why they at the beginning they choose this hypothesis because of this experiment. This experiment it produce enzyme, so they just directly there. But after that they make a discussion or they cannot ini tak betul, tak accurate, not accurate. So in conclusion, they conclude one gene, one polypeptide hypothesis, meaning that one gene will produce one polypeptide change. They conclude this. One gene will produce one polypeptide change. So these are all about the Bieder and Tatum experiment. Yang ni lah semua. So please read it. You need to know it. All of this experiment. Ini eksperimen prosedur dia lah. You need to know the procedure, result and conclusion. Kalau memang essay question dia come out, hopefully you can answer it. Okay, so that's all for this experiment. So this experiment conclusion is to express prove that our gene can produce protein. Prove that our gene can produce protein only. But how to produce, later you will learn. This experiment just want to prove that the DNA replication. DNA replication will prove that what is DNA carry the genetic material and what is the mode of DNA replication. And then this one is just proof that our gene material can produce protein. Only that. Okay, that's then we will learn about genetic code. Genetic code. So you will see this table. So if you want to ask me, I think I need to memorize all this or not? I think no need, but there are certain you need to memorize. Certain code you need to memorize, which is this lah. 3 this and also this. Yang lain tak perlu. Only these 4 code you need to memorize. So what are these? What are these genetic code? So genetic code actually is the genetic information that able to inheritance and store in DNA. It is the genetic information that can be able to inherit it and then can be stored in form of DNA. So in normally our genetic code will be expressed in mRNA one, not DNA form. Bukan dekat DNA format one. Not in DNA format one. We express in we will use in format in mRNA format. mRNA dah RNA format punya. Kami tak guna DNA punya. Why we use DNA better than RNA? Because the not all the DNA molecule can be able to act as the inheritable material. Tak semua, bukan semua, hanya certain je. Bukan semua DNA akan will be take part in inheritance one. Not all. Some maybe just control je. Maybe ada lain function dia lah. But not all. So for to accurate, we use RNA. More accurate use RNA. So this is the definition of genetic code. Okay, so now we will proceed to the characteristic of genetic code. So before that, actually you need to memorize these 4 genetic code, which is UAA, UAG, and UGA. And another one is AUG. So this one is very important in translation process. Later we will discuss it. Translation process, these 4 genes play the very important role in this process. So what are they? What is the translation? We will learn later. So I don't need. So now I want to explain about the characteristic of genetic code. So what are the characteristics? So there are 5 main characteristics you need to learn. So first characteristic is genetic code exists in triplet in nature. Meaning that it is exists in triplet nucleotide. Triplet nucleotide meaning triplet code will result in triplet code. So that is why that when you see this structure, see this diagram, see this table, you will find that all of them are arranged in triplet one. Semua dekat triplet punya. Memang triplet punya. Hanya triplet code je. Tak boleh ada 4 punya. 4, 1, 2 atau 5, 6 itu bukan genetic code. Hanya 3 je. They arrange in the triplet. So when they arrange in triplet, so they will give the result of 64 different combination genetic code. So all of these total have 64. Kami ada 64 possible genetic code. So in the world, all of the organism only have this 64 genetic code. Semua sama je. Tak kisah human ke, fungi ke, bacteria ke, virus ke, semua orang hanya ada 64 je. Bukan cakap human got 64 and then dog have 40. No, not like that. All of the organisms have this 64. So it is different assist in triplet, and then this genetic code will provide information for the specified basements of the amino acid. So this genetic code, what are they? They are very specified to each amino acid one. So that is why that when you see here, you see they are responsible to this one, right? FEHE, LEU, ini adalah short form untuk amino acid. FEHE, if I no mistaken, phenyl kan, phenylene, leucine, alanine, valine, alanine, tak lupa, serine, inilah semua ni, semua ni lah. So this is the short form of amino acid. So in exam, if MPN keluar short form ni, dia keluar short form ni, and then it want you to come out in full name, normally in structure question lah. Please, you need to know. Memang kena tahu. Or maybe in objective question, dia bagi short form, dia bagi short. Tapi in the option, dia bagi full name. So if you see that, if you think that dia bagi short form, dia bagi short form and then full name, you can compare it, but actually you cannot. Actually, if you don't know, you tak memorize, actually you will confuse one. Actually tak sama. Some of them is not same as the full name of the amino acid one. So in conclusion, actually you need to memorize the short form of this amino acid represent to the full name of the amino acid. So sepatutnya awak kena hafal. Sepatutnya. Because STPM not like university. So in university normally you don't need to hafal. University sangat baik. Dia akan bagi semua ni. Tapi in STPM dia tak bagi punya lah. Memang MPN dia tak bagi punya lah. Dia tak bagi, and then you need to know lah all these short form name, what are they. Memang kawan kena tahu. So please, dah tak tahu ke itu apa itu amino acid tu semua, please refer back to your CU semester 1. Tak kisah awak ada BTEC tak ITEC, please read again. Please read that what the type of amino acid, memorize all these 20 types of amino acid and their short form. So not only this CYS, there are also the one short form, another short form is only represent alphabet. But this one normally tak keluar lah. MPN tak kejam macam tu lah. Just you use the short only one alphabet, then you want to choose by the amino acid name lah. Dia tak kejam lah. Normally it will give you the short form with the 3 alphabet lah. So you need to memorize. I just want to inform you guys lah. Don't think that you don't need to memorize it. You need to memorize. Okay, so another characteristic is degeneracy. Please, you need to write this. Memang kena awak kena tulis di. This baru ada marker. Kalau tak tulis memang tak ada marker. So what is this? Meaning that degeneracy is the meaning that the same amino acid can be coded by more than one base triplet. But so meaning that if you see that actually, like I give the example of alanine. So alanine is coded by these 4 genetic code. So all of these 4 genetic code actually is coded with alanine. So GCU, GCG, GCC, and GCA are coded with alanine 1. So meaning that same amino acid degeneracy, meaning that one amino acid can be coded by more than one base triplet. Maksudnya satu amino acid mungkin ada banyak triplet code. Maybe AAA ke, AUC ke, ACU ke, ataupun GCU ke, semua ni lah. Maybe lah. So these are the concept. So however, only one type of amino acid is special is methionine. Methionine is very special. It only coded with one code only, which is AUG. Hanya dia aja. Yang lain 19 yang rest 19 amino acid, it is coded with more than one genetic code. Only the methionine is only coded with this AUG code only. Hanya ini je. Yang lain tak ada lah. Tak ada genetic code coded with methionine. Why? Because this one you need to know. Please remember because in translation process it is very important. It is very important one in translation process. Ini awak memang kena hafal. Okay. Next is about non-overlapping. Our genetic code is non-overlap. So, meaning that the same letters in genetic code cannot be used for other two codons. And then in one genetic code, I use laser. One genetic code, all the letters are unique and cannot be overlapped. This means I use this diagram more easier. So, if this is the codon, maybe codon meaning that this is the gene sequence, mRNA sequence that you see it. So normally in mRNA sequence there will not dia tak keluar macam ni lah. Actually dia tak akan keluar macam dia tak akan separate triplet you need to separate by itself. Actually if you see the gene sequence, the computer sequence one, dia akan keluar semua alphabet punya. Dia tak akan bantu awak triplet yourself one. You need to triplet yourself one. So normally if let's say these are the mRNA codon, so if you think that if you finally prove that these are the three genetic code, AUU is one genetic code, GCU one, CAG1. So for non-overlapping, so E is non-overlap one. So AUU, GCU, CAG, these are all the genetic code. But for this one, if it is overlap, meaning dia akan I use the example AUU, dia boleh overlap. Maksudnya U ni, tengah U ni mungkin dia is not for this code only. It may be can use for this code. Maybe U ni dia tukar kepada yang pertama. And maybe this U, maybe dia akan boleh tukar kepada yang ni. Maybe, and then this G, maybe juga boleh tukar kepada ini. It is overlap lah. Dia akan overlap punya. So actually genetic code is cannot overlap one. Yang ni we cannot say that this is genetic code lah. Because all of the character is unique. So if you use AUU, meaning these are the spatial one. Mereka tak boleh overlap guna yang lain punya. Memang tak boleh punya. Hanya ini je. Mereka hanya abang beradik je. Sama kawan je. Tak boleh kawan dengan orang lain punya. Memang kami tiga-tiga kawan baik-baik je. Awak tak boleh sesuka hati nak kawan kepada siapa punya. Tak boleh. You only can friend with me. You cannot friend with other. Kami tiga memang peluk-peluk together. We are the guys. We are the friend, the best friends. We cannot have the other friends you cannot go to make other friends. So this is what we call as overlapping. Another characteristic is universal. This one I already explained. But universal meaning that all of the organism, if you are a live species, live organism, you will use all these genetic code. Tak kisah awak siapa. Tak kisah you are the human, you are the plant, you are the animal, you are the microorganism, you are virus. All of these they use same genetic code unless you are the living organism. If you are not the living organism, then you will not use this code. Right? Okay. And then next is non-ambiguity. Meaning this one is that one genetic code will only code with one amino acid only. Yang ni terbalik lah. Tadi cakap apa? Tadi degeneration cakap one amino acid can be coded by many genetic code, right? But one genetic code is only can be coded with one type of amino acid only. Maksudnya kalau lah I use example AUG. AUG is only can be coded with methionine only. Dia doubly coded with other type of amino acid. Why? Dia doubly coded with maybe aspartic acid. Dia doubly coded with lysine, glycine, all this. Cannot. It only hanya boleh coded with methionine only. Macam boyfriend girlfriend lah. Bukan boyfriend girlfriend lah, ibu bapa awak. Macam ibu awak. AUG adalah anak untuk dia. Ibu dia AUG lah. Dia tak boleh ibu lain kan. Takkan lah methionine ada ibu lain because GGG. GGG bukan ibu untuk dia. Ini orang lain punya. So if you recognize your parents, specific lah. Takkan lah awak boleh pergi ke public ada nampak orang tua awak cakap itu ibu saya, itu bapa saya. Takkan. So every amino acid have their own parents lah, in general word lah. Have their own genetic code. One genetic code only can have one type of amino acid, but one type of amino acid can be coded by different type of genetic code. So please differentiate them. Please differentiate them. Jangan confuse. Okay. Okay, so that's all. So there are total 5 characteristics, right? 1 triplet, 2 degeneracy, 3 non-overlapping, and 4 universal, 5 non-ambiguity. So that's all for the part gene expression for intro part. So any question or no? I hope that my line is clear yet. I'm not share screen betul ke? Oh, okay. Oh, okay. Oh, tadi saya dah quick. Okay. Okay, so that's all for the intro part. So any question or no? No question. Okay, semua expert. Orang yang expert. Okay, never mind. So now we proceed to gene expression. To view gene expression, or another name we call as central dogma. Central dogma. Another name what are they? Actually, in simple word, that you will see the word, you will need to know the process of transcription and translation. These are the process of gene expression, transcription and translation process. So in general, in simple, transcription is a process to produce mRNA from DNA, and then translation is produce protein from mRNA. So what are the mechanism? What are the process? Later you will know it lah. Later you will learn it. So in general form, central dogma meaning is DNA to mRNA and then to protein. Okay. So for this one, I will explain about transcription. So what are the meaning of transcription? Meaning that it is the synthesis of mRNA using information in the DNA. Dia akan synthesis mRNA dari using the information in DNA. Okay. So in general, transcription process, we have 3 main process. Which is initiation, elongation, and termination. These are the 3 main process. Initiation, elongation, and termination. Okay, so what are they? We go to one by one. Initiation, what are they? Initiation, you can imagine that start of the transcription process. They need to start. Mereka kena start. But how to start? Where to start lah? Because in DNA replication, we know that DNA replication, how to start? It start from origin of replication, right? In DNA replication, it start on from origin of replication, and then it will unwind the double structure of DNA, right? Do you see here? DNA structure, right? DNA structure, and then it will unwind the DNA to form the daughter DNA cell, daughter DNA strand, right, for DNA replication. But for transcription process actually is same also. It is same also. It require many enzyme. It require enzyme also. It require all of them, all of the thing also, factor also. So initiation is how to start the transcription. Macam mana start? Siapa start lah? Tak, dia tak, bukan start sesuka hati. Oh, bukan ini ada DNA, saya start sini. Saya start sini. Bukan pilih punya. Dia pilih tapi dia ada specific location punya. So, initiation of this is prokaryote and then this is eukaryote. So, there are the differences. Ini ada perbezaan Ada perbezaan between eukaryote and prokaryote. Prokaryote is more simple compared to eukaryote. Okay, prokaryote 1. So for prokaryote, so there are one enzyme you need to remember is RNA polymerase. This is the key enzyme. For eukaryote also same, but eukaryote we will use RNA polymerase 2. Yeah, eukaryote is 2. Prokaryote is does not have 1, 2, 3, 1. Only RNA polymerase 1. Only one type. But for eukaryote, we have 3 types of RNA polymerase. But the enzyme that responsible in this reaction is RNA polymerase 2. The second type 1 and 3 does not responsible in this. There are other function lain. Tapi awak tak perlu belajar because you are not university level. Okay. So prokaryote meaning that RNA polymerase is the responsibility enzyme to it. So what are these functions? So if you see here, ini color lah, yang ni kuning ke? Skin color Skin color is all the RNA polymerase lah. So it will bind to the promoter first. Dia akan bind to the promoter region. This region we call as promoter region. Promoter region. Promoter region. Dia akan bind dekat promoter region. So, what are the function of RNA polymerase? There are many functions actually. RNA polymerase, there are many functions. Many function. First function is to unwind the double-stranded DNA. Dia akan untwist. Karena our DNA is double-stranded, right? So, dia kena separate, kena cut the DNA become single-stranded. And then, another function is to synthesis mRNA. Synthesis RNA, not mRNA. RNA nucleotide. These two major functions. Dia akan unwind double-stranded DNA and then synthesize RNA nucleotide. So, first, you need to know what are the promoter region. So, how the RNA polymerase tahu, oh, ini promoter. Kerana awak tahu, DNA sequence is very long. Also, kenapa dia RNA polymerase sangat pandai, dia boleh tahu. Macam mana dia boleh tahu? So, dia boleh tahu based on these two sequence. So, these two sequence you need to memorize. Either it is TTGACG or TATAAT. Ini memang kena memorize punya. Tak ada pilihan lain. Memang. Kalau dia tengok two sequence these two sequence, then it will start to bind it. And then it will initiate the initiation process, meaning it will start to unwind the double-stranded DNA structure and then start to synthesize RNA nucleotide. But synthesize RNA nucleotide is elongation part lah. For now, it is only unwind the double-stranded DNA only. Okay, so and then this this is for prokaryotic cell. For eukaryotic, same concept, but the promoter, the process is more complicated compared to prokaryote. Prokaryote is very simple. Prokaryote, dia tengok ada dua ni je, dia terus bind je. Terus bind and then terus buat reaction. But eukaryote is more complex. Dia tak senang seperti seperti prokaryote. Eukaryote is more complicated. So, eukaryote will also have the promoter region. the RNA polymerase will also recognize the promoter region. So for here, we will use RNA polymerase 2. So please differentiate prokaryote and eukaryote. Mereka dua tak sama punya, please. Okay, RNA polymerase 2. So they will use so the promoter region for eukaryotic cell is TATA box. TATA box. Why it use TATA? Because dia akan kebanyakan dia hanya ada TA sequence punya. Containing TA sequence. Kalau dia tengok dia ada TA sequence, dia akan start to bind. But before that, dia tak akan start bind. Why? Because dia akan ada several factor dulu. Dia ada several factor dulu. Because compared to prokaryotic, eukaryotic cells, normally the DNA sequence is very long. Sangat panjang compared to prokaryote. Prokaryote normally is bacterial, right? Bacterial, normally the DNA sequence is not very long compared to eukaryote. Eukaryote DNA sequence is very long. Like human, normally the DNA sequence, if you can separate, become a line, actually it can round around the earth, right? It can round around the earth 3 to 4 times. 3 can round around the earth, so it is very long. So it is very hard to only the RNA polymerase 2. When you see the tata box, it finally directly bind, cannot. Eukaryote tak boleh. Eukaryote is more hati-hati sikit lah. Dia ada several transcription factor, ada sedikit transcription factor. So normally this is either protein or enzyme. They will vagonize the tata box first. They need to vagonize. Is this confirm tata box tak? Maybe dalam mereka awak tengok jauh-jauh, dia macam tata box. Tapi awak pergi near, dia bukan tata box. Macam awak tengok lah Leng Loi, tengok dia sangat cantik, Leng Loi, handsome loh. Tapi awak pergi, apa ni tak cantik lah, tak lengkang lah. Ni semua 50, 60 tahun lah, bukan. Awak pergi jauh lah. Yang ni pun sama. So there are the transcription factor. This play the role to vagonize first. It need to vagonize the tata box first. Once it fully vagonized, ini memang tata box, must bind to the DNA before RNA polymerase can bind in correct position and orientation. Once it conform, bagi signal, dia akan bagi signal kepada RNA polymerase tu, ini boleh lah, ini boleh, ini memang okey punya. Okey punya. Ini dia macam awak pergi tengok, ini memang leng loi leng cai. Boleh. Ini memang boleh. Okey, lepas tu RNA polymerase tu baru datang by dekat promoter region. So our transcription RNA polymerase 2 will start with bind to the promoter region, and then will form the transcription initiation complex. So why it is complex? Because it not only have RNA polymerase 2, it also have transcription factor at there. And then it will unwind the DNA double helix structure, and RNA synthesis will start to begin. So this is more complicated for eukaryote. Pick again, prokaryote senang je. Dia ada dua region ni start directly bind. RNA polymerase, and then it include RNA polymerase. And then this one, dia kena ada transcription factor, vagonize dulu, kena bind dulu, baru RNA polymerase tu baru bind dekat promoter region. So this is differences between eukaryote and prokaryote. Okay, this is explanation. You can read yourself. And then next step is elongation. So this step, we not separate into prokaryote and eukaryote, all same. So semua sama saja. Yang ni sama sahaja dengan DNA replication process. Sama je. Just different is it add RNA nucleotide lah. Dia bukan tambah DNA. Dia tambah RNA. So if the complementary of the A is U lah, dia akan tambah U, bukan T lah. If the DNA is T, right, but for RNA is U. And then the differences between the DNA also, it produce RNA, right? And then Second difference is only one template. Only one DNA, one strain act as the template. One strain act as template only. So only one DNA strain, one DNA. DNA strain act as the template only. So normally, so if you see here, this one yang ni act as the template. Hanya ni je act as template. Yang atas ni 5 to 3 direction one, ini is also act as template. Dia dekat sana je lah. Tak buat apa-apa lah. Tak buat apa-apa pun lah. Hanya bawah ni je act as template. As template to copy the information so the RNA polymerase will add the RNA nucleotide to based on the complementary of the using the template, one of the template, and then the direction same as also 5 to 3 direction, same as the DNA replication. So normally the process is same, just the differences is it is produce RNA, dia tambah RNA nucleotide, and then only one of the template strain is act as the template only. DNA strain act as a template. So other strain is not as template dia macam tu je lah. Okay, so this is a elongation. So normally you can imagine addition of RNA nucleotide. So this is the main phase to synthesize mRNA to produce mRNA. Okay. After that, we will proceed. This is the explanation. You can read yourself. After that, we go to termination. Termination meaning terminate. Kami nak stop dia lah. Because we need to know mRNA is RNA. One of the difference between DNA and RNA is what? I think you still remember. RNA is more shorter, right? And then it is a single-stranded polynucleotide. Right, and then it is more shorter. So meaning that actually dia tak akan copy semua information punya. Tak akan. Dia bukan macam DNA. DNA is copy all the information. Semua information dia copy. Tapi mRNA dia tak copy semua. Dia hanya copy certain part only. So how to stop it? Macam mana? Siapa bagi signal dia stop? Kalau tak ada signal, memang dia akan produce seperti macam DNA punya. Tapi dia memang ada signalnya. So, termination is the transcription. Termination is occur when the newly formed mRNA reach the terminator. Dia akan reach the terminator, and then the enzyme RNA polymerase will be detached and mRNA move away from the DNA molecule. Okay, this is just a basic description. So, you need to detail between prokaryote and eukaryote. Prokaryote and eukaryote are totally different one. But the same as they will reach the terminator. But what are the terminator of prokaryote and eukaryote? For prokaryote, there are two types of termination that can maybe occur. One is intrinsic termination and another one is RHO dependent termination. So intrinsic termination is more easy. So intrinsic termination is when the DNA nucleotide contain inverted repeat sequence. Maksudnya awak tengok macam ni. Inverted repeat sequence, after that they will produce the same nucleotide already. You see here. Awak tengok ni, sini. So yang belakang ni semua tak sama. Semua A, yang ni semua T, semua ni. And then before that, there are the inverted repeat. It will have the inverted repeat one. Inverted repeat meaning that the sequence of this actually is same as the sequence of this. So if you read the sequence, this sequence and this sequence one, you will see that totally same one. If you read from 5 to 3 direction and 5 to 3 direction, T C G A C T G, you will see that T C G A C T G. So totally same. And then after that, they will continue to the repeated sequence. Or T, semua ni. So when they reach the RNA polymerase, reach the this inverted repeat sequence, and then it will start to form. Inverted repeat sequence. So these are the RNA mRNA that will be produced. Okay. So when it produce the RNA polymerase see that semua macam ni dah sampai ni, dia akan form the hairpin structure. The presence of the RNA hairpin structure. Why it can form hairpin structure? Because this actually is same as here. Yang ni mereka adalah why they are awak tengok ni. Awak tengok ni. Memang kerana because of this inverted repeat sequence, so that this both sequence ni are complementary to each other one. Because they are inverted repeat for in DNA. So of course RNA also has the complementary effect lah. So they can form the complementary they are complementary to each other, and then they can form bond, hydrogen bonding. They will form the hairpin structure. And then later it will followed immediately by a U-Rich region downstream will cause this one. Then RNA polymerase tahu dah sampai ni, okay, memanglah sampai terminator region lah, and then it will start to release the RNA polymerase lah. RNA polymerase will stop the transcription and then release from the DNA lah. This is one type. And another type, the explanation you can read yourself. And another type is RHO dependent termination. So this one also prokaryote, another one. This will involve one enzyme called RHO. This is one of the enzyme, RHO enzyme. Okay, this one is depend on RHO. Kalau bukan semua tu ada inverted sequence. Kalau dia tak ada inverted sequence, it will depend on the RHO. So normally, RHO accompany RNA polymerase and it synthesize the growing mRNA. So RHO actually is dia akan bergerak dengan RNA polymerase punya. Dia akan stay dekat belakang RNA polymerase to standby lah. Kalau dah sentuh, okay, dia start to have the reaction. So once the RNA polymerase make the RHO recognization site, RHO will start bind to this and cause a change in conformation of the RNA polymerase. Meaning that when the RNA polymerase reach the RHO recognization site, so normally the RHO recognization site is reached in C and G nucleotide. When it is reached in C and G nucleotide, normally is known as recognition site. RHO akan activated and then it will bind to the RNA polymerase. And then when it bind to the RNA polymerase will change the shape. Dia akan tukar shape dia. So it will pause the termination process because RNA polymerase already changed the shape. Maksudnya dia tak boleh function lah. So RHO then untwist the newly formed mRNA. So it need to untwist. Dia akan potong newly formed RNA from the DNA because DNA is still is directly stop. Dia memang macam ni punya. Awak tengok dia macam ni punya. Dia stop je. Kalau macam gerak macam ni, dia stop. Directly stop at all. Directly stop. Tapi masanya still ada RNA nucleotide still bound here. So it need to unwind this molecule. So it need to unwind this mRNA from the DNA and then form the mRNA sequence and then directly RNA polymerase will detach from DNA. And then also RHO will detach from mRNA. So this one, the differences between this intrinsic termination is intrinsic termination when it reach the inverted repeat sequence, baru dia start. But for this one, it is depend on the RHO termination. Okay, this one is about prokaryote. Next, we go to eukaryote. Eukaryote is more complicated. Actually, even more complicated. Got two diagrams. So eukaryote, actually there are many factors, many enzymes. So I don't know pun lah. If MPM keluar, awak nak explain macam ni tak? Saya pun tak tahu. Because if you refer MPM syllabus, it only say that want you to explain basic of translation. and you go to the Campbell, Campbell dia pun tak ada explain banyak tentang termination process pun, memang tak banyak. Campbell dia keluar 2 sentence ni je. Memang dia keluar dua sentence key sentence macam ni je. Tak ada lah. But actually if you refer further, actually it's more very detailed lah. So actually for eukaryote, actually it involve many enzyme. We can say that it involve many protein factor. Okay, it involve many protein factor. So how it's recognize the termination process? So normally when it have this signal, kalau RNA polymerase it give this signal AAUAA, normally is A AAUAA, meaning that afterward they are all of them will become A directly. So when they become AAUAA, the one of the reaction is called polyadenylation reaction signal will be activated. So this is the reaction lah. AAUAA, this is the all the reaction. What are the reaction occur is the all of this complex CPSF73. These are the protein factor. They will start to bind to this region, and then it will cut down the RNA string, directly cut down. And then this one, it will cause the RNA polymerase change the shape. Dia akan tukar RNA polymerase change the shape, and then there will another enzyme called isonuclease degradation enzyme. It will degrade the useless mRNA strand, and then detect RNA polymerase 2 will be detached from the DNA. This is just a general lah. If you want to see detail, you need to study the book in the genetic book one. A genetic book will explain more detail lah. I just explain in general je. So ini pun sama. So actually this is all about the transcription in eukaryote. Okay. So that's all for eukaryote. All about the transcription process. So any question? Tak ada question tak? Tak ada. Wah, semua expert tu. Semua tahu tu. Wah, sangat hebat. Okay, next I will go to the RNA splicing process. Okay, so RNA processing. Yang ni hanya special untuk eukaryote only. Only eukaryote only ada. Hanya eukaryote. Special for eukaryote cell only. Eukaryotic cell only. Prokaryotic cell tak ada proses ni. Why? Because eukaryotic cells, when we express the gene, we will have two regions, intro and exon. So what are the meaning of this intro and exon? So exon is the gene, is the codon gene that we want. It contains the codon that carry the inheritable material. Yang kami nak punya. It will express the protein amino acid one. But for intron tak ada. Intron tak berkait. Tak berkait. Tapi bukan tak cakap dia tak ada function langsung. But intron is the gene sequence that does not play responsibility in the production of amino acid. So in eukaryotic, why only say that eukaryotic only? Because eukaryotic, the DNA sequence are very long. Only eukaryotic, but prokaryotic is short, so it only have exon. So normally prokaryotic only have exon only. Dia tak ada intron punya. Semua gene yang express memang akan play the role in amino acid production. But for eukaryotic, no, eukaryotic have exon and intron. But we only want exon. So we will go to the RNA processing, we call as RNA splicing lah. RNA splicing to cut down all the intron to become the mRNA, real mRNA. For now we only have pre-mRNA, hanya pre-mRNA. Ini we call STPM BIO. Yang ni tak akan, we will not directly go to the translation process. We will not directly. Hanya yang ni to confirm that all the mRNA only have the intron, not intron, exon je baru boleh proceed translation. So what are the process of RNA splicing? So before that, if we want go to the RNA splicing, you will see that we will have to add two components at the end. One is cap at here, and another one is poly A tail at here. Kena tambah dua komponen ni baru boleh buat. Kalau tak buat, memang tak boleh. Ini dua komponen play the role. So what are they? So additional of cap at the 5 end. So what are the cap? So the cap is guanosine triphosphate GTP. Dia akan GTP will cap with the nucleotide. All of this nucleotide. Yang ni lah. Ini nucleotide ni. Dia akan connected to the nucleotide. So this is just lah. I want to show lah. Additional of gap. If you want to go to RNA splicing, you need to add this first. Kena add this. Kalau tak, memang tak boleh. And then additional of poly A tail. Okay, yang ni lah poly A tail at here. So why we want to how to add it? So normally this one will play will have the play the same as the eukaryotic. Dia terus sama dengan eukaryotic termination. So you see here, actually this one, all of these actually is in the termination step. Yang ni lah polyadenylation complex, yang ni adalah termination step 1. Yang ni termination step 1. Termination step 1. And then after when it recognize termination, it will cut down, cut the dia akan ini complex, dia akan cut the mRNA. And then after that, RNA is cut free. So now the mRNA is free now. And then this complex, also same as the fluorinylation complex, will start to add the A nucleotide. So who responsible is poly A polymerase lah. Di dalam ni ada satu enzyme we call as poly A polymerase. It will add the poly A nucleotide tail at there. Semua A punya panjang punya. So this is the process lah. So normally it will continue from termination 1. After termination, it directly go to this process. Okay, this is the explanation. Okay, next we go to the RNA splicing. So how RNA splice? Kenapa RNA splice? Kenapa kami nak tambah ni? Kenapa nak tambah ni? Tak boleh je, tak boleh meh. Directly splice, tak boleh meh. Memang tak boleh. Kenapa? You go to the mechanism. So RNA splicing, it involve also many factor we call as ribonucleoprotein. Ribonucleoprotein. So there are 6 types of ribonucleoprotein, U1 until U6, but U3 we does not have U3. We replace at U2AF. So why? I don't know. You don't want to ask me. You want to ask the scientists. The scientists that we name this. U1, U2, U2F, U4, U5, and U6. So these are the play the role. So how they play the role? Okay, so these are all. This is the process of the RNA splicing. Okay, firstly, our U1 and U2AF akan bind the U1 will bind to the 5' end splice site, and then another one U2AF will bind into the 3' end. U1 bind to the 5' and U2AF bind to the 3'. After that, U2 will bind to the branch site. Yang ni dekat sini. U2 branch site. Branch site. What is the branch site? This one, U2 one. At here. At here. Okay. So after that, later U4 and U6 bind to the U2. U4 and U6 will bind to the U2, and then a loop will form. once U4 and U6 bind to the U2, and then the loop structure will be formed. And then U5 at the same time, the U5 also bind to the downstream axon. Dia akan bind dekat sini. Lepas tu, after that, all of this complex will be form the loop structure. Dia akan tarik intron tu dia akan tarik move ke sini, and then UI will help to move ke sini to form the loop structure like this. Dia akan form the loop structure, and then after that our U1 and U4 will be detached. Dia akan dissociate, U1 and U4 will dissociate, and then U6 will replace the U1 punya tempat. Actually U1 dekat sini, apa lah. You see here U1 dekat sini kan. Lepas tu U1 detach and U4 also detach. U6 will replace this place, and then it will form this structure again, loop structure. Okay, next we go to the further. When it form this structure, actually this one is already it will directly to form the yang ni dia akan cut down, and then it will form bind to the branch side. So two axon and the intervening intron with their 5' to 3' side and the branch side are shown prior to the splice side. And then 5' side is cut first. Dia akan cut. Lepas tu U6 ni dia akan cut down, and then it will form this structure. The 5' side is cut first, and the free end of the intron looks back to the bind to the branch side. It will bind to the branch side. So yang ni lah, dia akan cut ke sini, lepas tu dia akan bind ke sini, bind to the U2 part. Bind to the branch side. After that, the 3' splice side is severed next, and the end of the two axons are joined. The intron is released as the labia structure. So if you see here, the 3' side is severed, dia akan cut down, and the end of the two axons will join. So meaning that yang ni and ini dia akan cut down the 3' end punya tempat. Lepas tu, after it's cut down, this axon 1 and axon 2 dia akan join together. So you form this structure, join together already. So that is why we want to have the poly A tier and 5-cat one, 5-cat structure. We need to to ensure that the axon is not cut down. Kalau tak ada ni, maksudnya kalau kamu tak ada add this cat, if let's say this one is axon, kalau kamu cut down, using RNA splicing, you cut down, so maybe you will cut down the gene in the exon. So kami tak mau, kami nak exon. So you cut down exon, apa nak kami nak lagi? So tak boleh. So that is why poly-A and cat is very important. So the intron is released as the labia structure. So it is released in form the labia structure tadi lah, macam ni punya. So mRNA is formed. So that's all. Any explanation you can give yourself. Okay, so finally our mRNA with only exon is formed and intron is removed. Okay, so that's all for the transcription process. Okay, any question? Ada apa soalan? No question. Wah, semua expert loh. Wah, memang expert kau. Okay. Okay, next I will proceed to translation process. Okay. Next is translation. Okay. I share the slide. Okay, you can see the slide, ma. Okay, you can see the slide. Okay. Okay, now we go to the translation process. So we already learned DNA to mRNA. Right. Transcription. So we already learn it. Right. Kami dah tahu lah mechanism dia, semua dah expert. So tak ada soalan kan? So memang orang dah expert. Okay. Now we go to the translation, which is mRNA to protein. Okay, mRNA to become protein. How does this occur? Okay. Okay, so translation meaning is synthesis of polypeptide using the information of mRNA. So before that, we need to know the ribosome structure first. Sem 1 with gap, Sem 1, semester 1. This one you already learned in Sem 1, right? So you should know. You should know already. If you don't know, mean that you not for you Sem 1 main-main. Kalau tak tahu memang awak Sem 1 main-main je lah. But you need to know structure of ribosome. So, structure of ribosome. So, if you know that our ribosome contain large unit and small unit, right? Large subunit and small subunit, and when they combine together to form the ribosome. And then there are 3 sites at here, 4 sites at here. A site, P site, E site, and also mRNA binding site. So, our mRNA will bind at here. Dia akan bind dekat sini. And then A site, P site, and E site is for tRNA. So you need to know in translation process there are many components. First is ribosome. Ribosome, tRNA, and mRNA take parts in translation process. Tiga benda ni. Ribosome, tRNA, and mRNA take part in translation process. So how it take part? So before that, we need to know the structure of ribosome first. So this is the structure of ribosome. So please know A site, P site, and E site because these 3 sites are very important. They have their function. So A site is amino acid tRNA binding site. P site is the bacteria tRNA binding site. E site is the acid site. In general, semua site ni adalah bind untuk special untuk tRNA. But what kind of tRNA we will proceed. Okay, so the ribosome structure. Okay, so before we go to the translation process first, we need to activate the tRNA first. Because mRNA to produce protein, it require the basic monomer of protein is what? Amino acid, right? So, macam mana nak tambah amino acid? Amino acid bukan tambah sesuka hati. Bukan tanpa sesuka hati. Bukan macam DNA nucleotide or using the template and then amino acid like A, U, C, T, G, and then amino acid sesuka hati tanpa ke sini. Oh, ini 1, ini 2, ini 3. Tak boleh. Dia bukan macam DNA. Bukan macam DNA punya. Kan? So, it is the activation of tRNA. So, you need to activate tRNA first. So if you know, please recap again your Sem 1. You learned about tRNA. You know that tRNA is the molecule, it will carry the amino acid, right? If you still know the tRNA structure, I don't know you guys know or not, dia macam ni kan? tRNA structure dia macam ni kan? And then from at here, we have the anticodon, right? Anticodon. And then here is the site of the amino acid, right? Dia akan bind dengan amino acid. Right? Right? I don't know. You still remember or not? Kalau tak ingat, please go back to your semester 1. Okay, I don't want to explain this more. So you know the tRNA structure, so dia macam ni. So you know that each tRNA will bind with the amino acid. And then there are anticodon. So this anticodon is responsible to determine what kind of amino acid want to bind it. So okay. So actually our tRNA actually initially does not bind to an amino acid. We need to activate it. Baru dia boleh bind. Kalau tak boleh bind punya. So that is why we call as activate tRNA. So how it be activate? We need to have one enzyme called triosine tRNA synthetase. This enzyme, okay. So firstly, there are tRNA and also amino acid. Responsibility are amino acid. For this one, this book Campbell dia guna AUA. So AUA is tyrosine. Responsible yang bind punya adalah tyrosine. So tyrosine and DNA will bind to this enzyme. Okay, after that they will catalyze the reaction. It will and consume ATP, catalyze the reaction, catalyze the covalent bonding of amino acid to its specified tRNA. So it will form the covalent bond at here. Okay, after that the tRNA charged tRNA. So this one we call as charged tRNA. tRNA is formed already, and then it will release from this enzyme. Triosyl tRNA synthetase. It will release from this enzyme, and then so this one is what we call as it is charged tRNA, which is already activated. They contain the respectively amino acid. So these are the activation of tRNA. Okay, so these are explanation. You can read yourself. Okay, initiation process. Okay, what is the initiation? So in the translation process, actually same as the transcription, we also have initiation, elongation, and also termination. Sama aja. Sama aja. So initiation, same as transcription, we need to know where to start. Mana start? How to start it? So that is why I say that in genetic code, you need to know AUG. AUG is as start codon. Start codon. Start codon meaning that it is the starting point of the translation process. Akan start dari sini. Memang semua akan start dari sini. Memang start dari sini. Dia akan start dari tempat lain punya. Okay, so AUG. So AUG is important. So you need to mention it initiation process. So actually in initially our first step is our mRNA will bind to small subunit of ribosome. They will bind to the small subunit ribosome first. The last subunit not come yet. Datang lagi. Dia akan bind dekat sini first. Okay. After it bind, lepas tu our bacteria mRNA binding site of this subunit recognize a specific nucleotide sequence on the mRNA sequence. Okay. And initiator tRNA will with the anticodon UAC base pair with the start codon AUG. So after that, the amino tRNA dia akan cari lah based on the anticodon here. So it will try to find lah. Which one is AUG saya nak cari AUG siapa? Saya adalah UAC. Saya UAC kan. So kawan saya adalah AUG. So AUG siapa? Oh, finally saya cari awak. Okay. After that, they will bind together. Dia akan cari start codon mana. Oh, finally dia cari. Okay, dia will bind to it. Afterward, it bind, it will consume GTP molecule, and then it will the last ribosome subunit will bind to it again. It will bind to it again, and then this molecule, this charged tRNA will bind to the P site. So the arrival of the charged ribosome unit complete the translation initiation complex. So this name kalau structure question dia cakap ini apa? This one is translation initiation complex. So protein co-initiation factor are required to bring all the translation component together. Hydrolysis or GTP provide the energy, so called hydrolysis GTP, and initiator tRNA is in the P site. The A site is available to the next amino acid. So P site dia akan directly dekat P site. What are the function of A site? Dia akan terima tRNA yang lain to form the amino acid, amino polypeptide chains lah. So this one I will explain in elongation. So in the explanation you can build yourself. Okay, elongation process. Okay, so for elongation process is more complicated. Okay, for elongation process, if you see here, this is the first process, second, third process. So if you see here, after that, our ribosome will start to move. Dia akan bergerak dia akan bergerak ke sini. Dia akan ke sini A side. Okay, dia bergerak ke sini. Okay, it will recognize. Ini apa? Maybe ini adalah UCG. Lepas tu anticodon dia memang kena ada AGC. So this is the anticodon from the tRNA. This is the mRNA one, codon from mRNA. So the anticodon tRNA with the already activated charge tRNA with the respectively amino acid for example like AGC, they akan start to bind to the A side. It will start to bind to the A side. Okay, so the anticodon or incoming amino acid base pair with the complementary mRNA will bind to the A site, and then got hydrolysis ATP. Many never are present, but only the one with yang ni last sentence ni just say that actually there are many type of activated tRNA at the surrounding lah. Banyak tempat lah, not only one. Actually got banyak to standby. Dia akan standby punya. But only the complementary to the codon or mRNA tRNA will bind into the A site. Okay, and then it will require energy. Okay. Require energy. So after that, the second is peptide bond formation. Okay, next is the polypeptide chain in the E side. So you will see that there are already amino acid here, polypeptide chain here. So this amino acid in the P side that bind to the tRNA one, it will form the peptide bond with the A side punya amino acid. Dia akan form the peptide bond at here. So optic peptide bond at here. So if you see the explanation is very round, the rRNA molecule of the large, you know that our large subunit of the ribosome contain rRNA, right? I think you should remember. Because you already learned in sem 1 what are rRNA. Okay, rRNA in the ribosome already in the large subunit will catalyze the formation of peptide bond. So this is one of the function of rRNA. So that is why that when you learn sem 1, the function of rRNA is also responsible in protein formation. Inilah function dia, the specific function lah. So actually for those who get sem 1, maybe you can write this out in your exam. Actually this is the specific function of rRNA lah. RNA molecule of the large ribosome unit will catalyze the formation of the peptide bond between the carboxyl end of the. So this want you to tell you that it form the peptide bond lah at here. Dia akan form the peptide bond. Okay, next. After it formed the peptide bond, because it already formed the peptide bond at here, meaning that the original bond at here, that bond with this tRNA at the P side one, dia akan break down lah. It will break down lah. Dia akan break down, and then rejoin, and then pergi ke A punya. Because it already formed the bond lah. It already formed the bond at here. So dia akan pergi ke A punya part. Dia akan pergi punya A punya part. After that, okay, so that is why we see that when you see as this step, remove the polypeptide from the tRNA in the P side and attach it to the amino acid to the tRNA in the A side. So in explanation, please mention carefully your side, A side or P side. Memang kena mention carefully. If you don't mention, you just mention tRNA, examiner nak cakap, tanya awak, side apa? Tempat mana? P atau P? A or P? Saya tanya awak A or P? Lose mark. Although your theory memang betul, tapi not clear. Okay, not accurate. So wrong. Okay, next. also will use energy UTP. After that, it will go toward translocation. So this process is that our ribosome will translocate the tRNA in the A site to the P site. So at the same time, so meaning that our P site punya tRNA dia tak ada amino acid, dia akan pergi ke E site already. And then at the same time, our A site punya amino acid dia akan pergi ke pergi ke P site. So you see this diagram, A site pergi ke P site. And then P site punya originally tRNA dia akan pergi ke E site. And then E site dia akan keluar. Keluar dari ribosome. So the mRNA move along with its bound tRNA, bringing the next codon to be translated into the A site. Okay, so the tRNA here dia akan keluar lah, tak guna lah, keluar. Lepas tu dia repeat again the process. Repeat again, repeat again, adding the tRNA and then repeat all the process until it can form the very long polypeptide chain lah. So normally it will form very long punya polypeptide chain lah. Dah sangat panjang punya lah. So until it reach, so this explanation you can read yourself. Okay, until it reach the termination process. Sampai termination. Okay, so when it reach the termination process, so it want to stop lah. Kerana mRNA although it's short, but actually not all the information will be read at all. So maybe some part dia akan stop. So how to stop? There are the codon responsible is stop codon. Yang ni lah awak memang kena hafal. Please hafal. UAG, UAA, and UGA. Kalau the translation process, the ribosome baca, dia baca ini codon. UAG, UAA, or UGA. Okay, after it reach that, tRNA dah tak boleh tambah ke A site lah. Tak boleh lah. Kenapa? Kerana ada satu factor we call as release factor, protein factor. Dia akan elongation terminal require GTP allosteric as well as additional protein factor. Okay, release factor. So this is the release factor. So when the ribosome reach a stop codon on mRNA, the A site of the ribosome will accept the release factor. So it is the factor, you can say that it is like the inhibitor lah. Lah macam inhibitor, but it is not inhibitor, it is release factor. Dia akan bind to the A site and then to stop the tRNA to binding. So the tRNA dah tak boleh bind lah. Okay, after that, the release factor will promote the hydrolysis of the bond between the tRNA in the P site and the last amino acid of the polypeptide. So meaning that this release factor will hydrolyze this bonding. It will break down this bonding, and then it will release the polypeptide chain. Akan release the polypeptide, and then this tRNA will at the P site will enter to the E site, and then it will release out lah, and then afterward the ribosome also will be separated. And the mRNA also be detached also. So the free polypeptide, our free polypeptide chains or protein molecule is formed now. And then this polypeptide will enter mana? Ingat? Mana? Will enter mana? Chapter 2 1, semester 1, chapter 2 1. Akan pergi mana? Pergi ayah. Kan? Aloi. I hope that everyone remember lah. After pergi ayah, pergi mana? Golgi apparatus. Lepas tu pergi mana? Dah protein lah. Dah protein lah. Tak boleh pergi mana-mana tempat. Inilah whole proses. Semua proses ni. So, senang kan? Saya rasa senang. Kenapa saya rasa senang? Kerana tak ada orang tanya soalan. Kan? Tak ada orang tanya soalan. So, actually our class is end already lah. So I already explained all about the gene expression. So okay, so that's all for class today. So any question? No question. Wah, saya rasa semua expert tau. Memang semua ni Semua orang yang paling expert lah. Harap yang join kelas ni semua boleh score lah. A lah. Dapat A semester 3. Should be. Because no question. Meaning that you all already very sangat cermat lah. Very smart people. No question at all. Okay, so that's all class today. So no questions. So thank you for attending the class. So see you tomorrow. So tomorrow we will proceed to the regulation of gene expression. This is the seventh topic lah. Finally seventh topic, and the last topic mutation. So I don't know I can finish tomorrow. I hope that I can finish this chapter lah, 17, chapter 17. Because it already takes almost 1 month. Almost 1 month. So actually it's very long chapter lah. And I hope that if you guys join from the first topic until now, my explanation give the beneficial to you all lah. So that's all for today. Thank you. Bye. Have a nice day. Good night.